According to the recent international diagnostic criteria for neuromyelitis optical spectrum disorders, anti-AQP4 IgG is considered to be a core diagnostic biomarker. Anti-AQP4 IgG detection by cell-based assay can facilitate clinical diagnosis. The cell-based assay is more sensitive and specific than other detection methods, and it can be applied to both clinical diagnosis and scientific studies.
First, bring the patient's serum samples, the biochip slide, the PBS powder, and the tween 20 from the refrigerated cell-based assay kit to the room temperature. Then prepare more than 200 milliliters of PBS wash buffer containing 0.2% tween 20 for each biochip slide. Use the wash buffer to make 10-fold serial dilutions of each serum sample. Prepare positive and negative controls according to the manufacturer's instructions. Add 30 microliters of 10-fold serial dilutions of the sample, the positive control, and the negative control to 5 individual reaction fields on the reagent tray. Hold the biochip slide by its sides and remove its protective cover without touching its reaction fields. Put the slide on top of the reagent tray and incubate the setup at room temperature for 30 minutes. Gently rinse the biochip slide with the wash buffer. Then immerse it in a 500 milliliter beaker.